We talk a lot about our DNA, the A's, T's, C's, and G's that make up our genetic code. But what if I told you that's only half the story? Beyond that code, there's this whole other layer of information, a kind of biological software that tells our genes when to turn on and when to shut off. And this system is never, ever more important than at the very start of a new life. So today, we're diving deep into the world of epigenetics and its fascinating connection with assisted reproduction. Here's our game plan. First, we'll break down what epigenetics really is using this idea of the genome's software. Then, we'll get into a wild set of parental rules called genomic imprinting and the incredible reprogramming cycle that goes with it. After that, we'll see exactly how reproductive technology fits into this picture, take a look at the data on some associated disorders, and finally, wrap up by talking about what this all means for the future of fertility.
All right, let's dive right in. What are we actually talking about when we say the software of the genome? We often think about our genes as this fixed blueprint, right? Like our destiny is written in our DNA. But is it really that simple? The sequence of your genes is, of course, fundamental, but what if the way those genes are used, which ones are read and which are ignored, is just as important as the genes themselves?
And that's where epigenetics comes in. It's this extra layer of instructions that runs on top of your DNA. Think about it. A muscle cell and a liver cell have the exact same DNA, the same hardware. So why don't they do the same thing? Epigenetics. It's the software that tells the muscle cell to act like a muscle and the liver cell to act like a liver by controlling which genes are active.
So how does the cell pull this off? It has a few key tools in its toolkit. First up is DNA methylation. This is like putting a tiny chemical off switch directly onto a gene, which basically tells the cell's machinery to ignore it. Then there's histone modification. DNA is wrapped around these proteins called histones, and by tweaking them, the cell can either pack the DNA up super tightly to hide genes away or loosen it up to make them easy to read. And finally, you've got these little RNA molecules that can jump in and fine-tune gene expression. It's an incredibly elegant system.
Okay, so now that we have the basics, let's look at how these tools are used in a process that is absolutely critical for reproduction, something called genomic imprinting.
Now, for most of our genes, we use both copies we inherit, one from our mom and one from our dad. They both get a say. But for a special group of about 200 or so genes, imprinting means we only use one of those copies. The other one is silenced, and it's all based on which parent it came from. The gene's origin story, its heritage, determines whether it gets to speak out.
So how does that work? This diagram really lays it out. Look at the sperm on the left. It brings in an active, unmethylated copy of a gene. But the egg on the right brings in a copy that's been tagged with methylation, that's the black bar, and that tag silences it. So after fertilization, the new embryo keeps that pattern and only the dad's copy is expressed. Getting this dosage, this 1-to-0 ratio, exactly right is absolutely essential for development.
But why? Why on earth would this system even exist? The leading theory is called the parent-offspring conflict hypothesis, and it's a wild evolutionary idea. It's basically a biological tug-of-war. The theory goes that the father's genes want to push for the biggest, strongest fetus possible to ensure his genes survive, even if it takes a huge toll on the mother. But the mother's genes need to conserve resources so she can survive the pregnancy and maybe have more children later. Imprinting is the molecular battleground where this ancient conflict plays out.
Okay, so we know what imprinting is, and we have a theory about why it exists. Now let's get into the how, the unbelievable process of how these imprints are set, erased, and then reset generation after generation.
This diagram shows the whole journey. It's a continuous cycle. In the body's normal cells, those parental imprints are carefully maintained. But in the primordial germ cells, the ones that will one day become sperm or eggs, the slate is wiped completely clean. Those imprints are erased. Then as those cells develop into either sperm or eggs, a brand new sex-specific pattern is put down, all ready for the next generation. It's a massive, genome-wide reset, and it has to be perfect.
So we can really break this whole dance down into 3 critical phases. First, there's erasure, wiping the slate clean in the future sperm and egg cells. Second, establishment, where the new made from mom or made from dad tags are put in place. And third, and this is so important, is maintenance. Right after fertilization, the whole rest of the embryo's genome gets wiped clean, but these special parental imprints have to be protected and carried through. It's an incredibly delicate operation.
And this delicate, perfectly timed process leads us right to the heart of our discussion today. What happens when we,
with our modern assisted reproductive technologies, or ART, step into this incredibly sensitive window of epigenetic programming. This diagram here shows the overlap, and it's pretty stunning. Procedures like IVF, ICSI, growing an embryo in a dish, they happen exactly during this period of establishing and maintaining those first crucial imprints. This is what scientists call the critical window. It's a moment when the epigenome is wide open, making huge changes and potentially more sensitive to its environment.
So what kind of environmental factors are we talking about? Well, researchers are studying things like the hormones used to stimulate egg production, the specific nutrients in the lab media where embryos grow, the stress of freezing and thawing, and even the physical act of ICSI itself. Each of these is a variable that's different from a natural conception, introduced right during that sensitive programming period.
Which, of course, brings us to the data. What do we actually know about the connection between ART and these rare disorders that are linked to problems with imprinting?
The main conditions that have been studied are things like Beckwith-Wiedemann syndrome, which causes overgrowth, and Silver-Russell syndrome, which involves growth restriction. There's also Angelman syndrome, a neurodevelopmental disorder. And the key thing here is that all of these conditions can be caused by mistakes in those epigenetic tags we've been talking about.
Okay, now, when we look at the numbers for Beckwith-Wiedemann syndrome, some studies have found an association that points to a relative risk for children conceived with ART that could be up to 10 times higher than for those conceived naturally.
But, and this is probably the most important point, context is everything. Beckwith-Wiedemann syndrome is incredibly rare to begin with, affecting maybe 1 in 15,000 births. So even if you multiply a very, very tiny number by 10, you still end up with a very, very tiny number. The relative risk might be higher, but the absolute risk, the actual chance of any one child being affected, remains extremely low.
And the science here is also incredibly complex. Just take a look at this table on freezing or vitrification. In mice, scientists found some epigenetic changes after freezing embryos, but when they do similar studies on donated human embryos, they often find no difference at all. This is a perfect example of why you can't just assume what happens in a mouse will happen in a human. It shows just how challenging this research really is.
So, where does that leave us? When you put all of this together, what does it mean for the future of fertility and the millions of people who rely on these amazing technologies?
You know, this quote from the source material really just says it all. More than 8 million healthy children have been born because of RAT. That fact alone is this powerful testament not just to the success of the technology, but to the incredible resilience, the plasticity of the human embryo itself.
So, the goal of all this research isn't to raise alarm bells, it's the opposite. It's about gaining a deeper understanding. By figuring out the precise molecular details of what's going on, scientists can keep tweaking and refining every single step of the ART process to minimize even these very small potential risks. It's about making an already safe technology even safer.
And that leaves us with this final, crucial question. As our technology gets more and more powerful, allowing us to participate in the very first chapter of life, how do we make sure we are doing everything we can to protect and support those very first, most fundamental instructions? It's a question science is working hard to answer every single day.